Xenotransplantation is a feasible method to treat organ failure. However, effective monitoring of the immune rejection of xenotransplantation is a problem for physicians and researchers.
This protocol is a simple, convenient, low-cost, and less invasive method to monitor the immune rejection of xenotransplantation.
This technique can be used in xenotransplantation field, including pig-to-human islet transplantation, kidney transplantation, liver transplantation, and heart transplantation.
Begin by transferring 500 microliters of blood samples into different microcentrifuge tubes. Add 20 microliters of protease K and 500 microliters of lysis buffer into the tubes, then thoroughly shake them to mix. Put the tubes in a water bath at 56 degrees Celsius for 10 minutes, shaking them 2 to 3 times during the incubation until the solution becomes clear. Centrifuge them briefly to remove the liquid beads from the inner wall of the tube covers. Then add 500 microliters of anhydrous ethyl alcohol and shake thoroughly. Transfer the mixture into the adsorption column. Then centrifuge the column for 2 minutes and discard the waste liquid. Add 800 microliters of rinse solution to each adsorption column and centrifuge for 1 minute. Leave the columns at room temperature for a few minutes to dry the remaining